but sometimes there are genes that are on this strand, sometimes there are genes that are on this strand. Okay? So in which case the coding strand can be this one or the coding strand can be this one, depending on which strand the gene is on. So coding strand and template strand is not really one strand of the chromosome. It can be different. For all genes, actually. Okay, so don't do that by itself. The cell needs, of course, a protein that is an RNA polymerase that will actually use the template strand to actually synthesize the RNA molecule. And of course, you have incoming ribonucleoside that will triphosphate, so this is ribonucleotide, that will be added to the elongating chain. And of course, the 5' end is the first end that is synthesized, and just like in DNA synthesis, the 3' end is being elongated. That's the very simplistic situation. Okay? Let's go into a bit more details. This is a bacterial chromosome, if you want, where we have one gene here to there, and from there to there. And what you see here, this looks like a Christmas tree. It's actually many RNA molecules that are synthesized in the same time from the same gene. So you should not see it happening as you have one gene and one RNA molecule is made, finished, a new one starts, finished. No, everything is happening at the same time, and many molecules of RNA can be made from one single gene. And what you have here, this black particle, is believed to be ribosomes that are already assembling at the 5' end to start translation of the messenger RNA. All right. Cells produce various types of RNA. In eukaryotic cells, this is what you have. Messenger RNA, they code for protein. You have ribosomal RNAs. They form the core of the ribosome structure and catalyze protein synthesis. You have microRNAs. They regulate gene expression. They do not code for protein. They regulate the expression of other genes. You have transfer RNAs. They actually serve as adapters between messenger RNA and amino acids during protein synthesis. So ribosomes actually use transfer RNA to translate messenger RNA. And then you have other non-coding RNAs. Now there are many types known, like long non-coding RNA, PIWI RNA, many types of non-coding RNA which mostly have a regulatory function. They regulate RNA splicing, they regulate gene expression, they regulate telomere maintenance, and many other processes. All right.
MicroRNAs transcribed from DNA?
Yes. MicroRNA are transcribed from DNA, and it took a long time for them to be known. Of course, they have always been there in our cells, but it's relatively new because they are extremely small, 20, 30 base pair. So because of their very small size, it took a lot of technique to actually detect them. So this is why it's kind of new. But they are from RNA, from DNA. Signals in DNA tell RNA polymerase where to start and finish transcription. This is what happened in bacteria. Very simple case where you have one gene, and on the one end, on the 5' end of that gene, there is a transcription start site, which is in green here. And there is a terminator. Okay. The promoter is the transcriptional start site, and the terminator is where the transcription will stop. Nothing to do with Arnold Trout again. And what happens is, in bacteria, there is this is the RNA polymerase here, and there is one small protein called the sigma factor that will recognize the sequence of the promoter. Then it will recruit the RNA polymerase and start transcription. So once the transcription starts, it's only the RNA polymerase that will move along the gene. The sigma factor will be released and can bind to another promoter, either the same one or a different one. So the polymerase moving down the gene will, of course, synthesize the growing RNA transcript. Once it reaches the terminator, of course, it will detach from that gene, and sigma factor can again bind to the RNA polymerase, and a new cycle of transcription can start. But there is a problem here. It's that there is a promoter here, so actually the RNA polymerase could very well bind here and go down there. But if you think about it, it could also bind here, but actually transcribe in the opposite direction on the opposite strand. Right? If you have a promoter here, it could be bidirectional. It could just go there, or it could just go there. How do you think this is achieved? There is no gene. Okay? This is the case where there is no gene. There is only one gene here, and that is the promoter, and that is the terminator. So how could you imagine a mechanism in which the cell could actually tell the RNA polymerase, okay, you have to start here, and you have to elongate the RNA molecule that way? Can you imagine a very simple mechanism in which direction is given to the RNA polymerase?
Maybe sigma factor has a kind of two guide RNA, and sigma factor can bind many specific direction.
Although guide RNA is the wrong word. Sigma factor has no guide RNA. Sigma factor is just a protein. Okay? But you are right in saying that actually sigma factor recognizes a certain structure in the DNA and bind a certain sequence. And because that sequence is usually that way, so imagine this is the start site here, so this is where transcription will start, and here this is the promoter. And usually in bacteria, promoter is two short sequences that contain usually a lot of T and A, especially TA sequence, hence the name, it's the Tata box. And this also exists in eukaryotic cells. Because of this sequence, this gives an idea of the direction. So when the sigma factor binds there with the RNA polymerase, it knows that it has to go in this direction because the Tata box is here and the other sequence is there. So it's that way it must go. All right? So the start site is here. So the RNA polymerase will use the template strand to synthesize RNA molecule that has the same sequence as the coding sequence. Terminator is the same. Terminator is also usually 2, 3 sequences that are organized in such a way as to give direction. Although it is less important here because direction is given by the promoter, so transcription can only start in that way. But as you can see, the difference between the promoter and the terminator is that promoter has a lot of T and A, the terminator has a lot of GC. Okay? And a very easy question is, why would that be? Why do you think for the promoter you have a lot of TA and for the terminator you have a lot of GC? Why do you think this might be based on the structure of DNA? In the past, I've asked a similar question about the where DNA synthesis starts. The origin of replication. So it's the same reason here. Because terminator is so GC rich, actually the RNA polymerase has a lot of difficulty to actually pry these two strands apart because they are very tightly attached to each other because of many hydrogen bonds between C and G. So this is why it is thought it's very efficient terminator. Okay, so that's the case in bacteria. Very easy. RNA polymerase, sigma factor, promoter, terminator. Fine. But of course, as I said earlier, one gene can be on different strands. So in this case, you have the gene A, which is on this upper strand here. So the promoter is there, the terminator is there. But on this DNA molecule here, shown here, then there is another gene who is on the other strand here. And in this case, the promoter is on the right, but it gives leftwards direction. Okay. So this RNA transcript from gene A and RNA transcript from gene B. So in this case, that is the template strand for gene A, and that is the template strand for gene B. Okay. That is the coding strand for gene B, and this is the coding strand for gene A. So you cannot say this is always the coding strand, this is always the template strand. It really depends on which strand the gene is. Okay, sequence is very important. Next point is the initiation of eukaryotic gene transcription is a complex process. So I want you to compare the situation, which is very simplified, with the case that we have seen in bacteria. First of all, in eukaryotic cells, there are three different RNA polymerases. There is the RNA polymerase 1, which is a protein that is specialized making RNA. It will transcribe genes coding for ribosomal RNA. There is the RNA polymerase 2, which is the polymerase that transcribes all protein coding genes, microRNA genes, plus genes for other non-coding RNA. Okay? For example, as we see before, RNA that are involved in the splicing of messenger RNA, which is something that we are going to see later. And then there is the RNA polymerase 3, which is specialized in making tRNA, also making one type of ribosomal RNA, and genes that code for other many small ribosomal RNA. So, in bacteria, there is only one RNA polymerase. In our cells, there are three. Okay. Usually, our cells have developed much more specialization than bacteria. But maybe the most obvious difference is that here, RNA polymerase is regulated by many different proteins. And there are proteins that are always there, and there are proteins that are not always there. So first we are going to see the proteins that are almost always there, okay? They are required to start transcription. And these proteins are called the general transcription factors. And that's the thing. So you can see this is very similar to bacteria in that you have the data box and then you have the start of transcription. First of all, you have a data box binding protein here, which is a subunit of a transcription factor 2D that binds to the promoter. Once this is bound, it recruits another transcription factor 2B, which binds to the promoter. Then this recruits the RNA polymerase that has many other factors on it associated with the promoter, and then you have two other general transcription factors coming, TF2E and TF2H. And TF2H is very important, not only because it's the subunit, actually it's made of different proteins, okay? But it is the subunit of the whole complex that will actually pull the two strands of DNA apart. And it also has phosphorylating activity. So it will phosphorylate the RNA polymerase and allow the RNA polymerase to actually leave the promoter and start the transcription. And something that is very important in eukaryotic cell, this is the tail here of the RNA polymerase. It contains many amino acids that can be modified, that can be phosphorylated to regulate its activity. Let's make it clear that all these transcription factors, they are only required for the recruitment of the RNA polymerase, not for the activity of the RNA polymerase. Okay? Once transcription starts, the RNA polymerase will move by itself. All right? And why this is happening? Because you can imagine the DNA is a very long molecule. Actually, what happens is that when the Tata binding protein binds the Tata box, it actually causes a kink in the DNA. So the DNA will be bent at that location, which provides a very specific structure that is then recognized by all the general transcription factors. In this case, of course, for simplicity, it's not shown, but you can imagine here there will be a really sharp bend in the DNA, and this makes a good structure for binding. And if you want to make analogy between what happens in bacteria and what happens in eukaryote, then you can say that the sigma factor here is maybe a little bit similar to the Tata box binding protein. It's a little bit similar. The role is a little bit similar. Okay. So that's just a fun film as usual, showing. You can see here the kink that is caused by the binding of the Tata box binding protein here. And because of this structure, then other factors will come. Recruit RNA polymerase, phosphorylate RNA polymerase, and RNA polymerase will start and synthesize RNA. So the protein should come. There you go. This also binds DNA. Many other factors come into play. And then that's it.
What was that?
That's necessary. So that you have the RNA polymerase with pulling apart the two strands here, and you have RNA with kind of coming out of the protein. Remember your question. Remember your question. RNA polymerase is much faster than DNA polymerase because actually it's not that important to make mistakes. Because if it makes mistakes, maybe only one molecule of RNA will have a mistake, but many other molecules of RNA will actually be all right. So actually, it's less important than DNA replication. And you cannot really afford to wait. If you need an RNA to make protein for a certain function, you cannot really afford to wait for many genes. It has to be made instantly, so this is very important for RNA polymerase to be fast. And then once RNA polymerase reaches the terminator, then it will stop. All right. But what we just mentioned, it's only the proteins that are required for almost every transcription. There are many other things. There are many other things that regulate the activity of the polymerase. So what do you think is this? Can I go forward? I wonder if I can go forward. Funny, there is no menu. But what is this? What do you think this? So not this one, wait.
A monster.
It's a monster. The very last thing is very difficult to see. So this is transcription factor 2b probably. And then other factors will come. RNA polymerase will come. And then there is one thing, RNA polymerase, that's the blue thing here, who came earlier. And then something will happen there.
Yeah, L.
What do you think this is? Based on what I explained before, what do you think this is? That you have this bit of DNA that comes. And what are the possibilities? What could it be? Based on this.
It looks like it's the same strand, which is like.
Yeah. So it could be different things. It could be different things. But based on this information, for example, based on what I told you, what could be this last thing coming? Now, when it comes directly, the RNA polymerase starts. Based on this, what could it be? It could be transcription factor 2H. Because this transcription factor 2H is actually the one that will finally phosphorylate the RNA polymerase and prompt it to start. So this is what it could be. Okay? This is the final thing that is required for RNA polymerase to start. This is what it could be.
Was it connected, attached to DNA?
What it could also be, and this is, I wonder if there is a picture. This is, unfortunately, it's in the next chapter, but for eukaryotic genes, the promoter is much longer. What is showing, what you see here, this is really the basic promoter. It may be only, like, for example, Tadabox, it may be what? 10, 20 nucleotides. Very, very short. But eukaryotic promoters are much, much bigger. They can be 1,000, 2,000, 3,000, even 10,000 kilobytes. And sometimes what you have is that regulatory transcription factor will bind upstream of the gene. It can be 1 kilobase, as I said, very, very far away from the gene. But because of the DNA being such a flexible structure, they can actually be brought together to promote the transcription of a gene. And in this case, so people will say that there is a lot of junk DNA that is not coding for protein. Actually, that is not true. DNA can also be used as a tether. As a tether for these many different transcription factors that regulate transcription to be brought together by folding of the DNA. So this is what's happening. So even if your transcription factor is very far from the transcription and start site, it can actually be brought together with the transcriptional machinery here and regulate gene expression. But it's not all. This is only transcription. Eukaryotic messenger RNA are processed in the nucleus. So we are going to see RNA processing. Yes, because if you think about it, this is a nucleus. And you have, inside the nucleus, you have a nucleolus. And this is where ribosomal RNA are being transcribed. But you have a double membrane here. You have the nuclear envelope. And you can see there are pores here. One is here, one is here, maybe one here. RNA is transcribed in the nucleus, but it's translated in the cytoplasm. So RNA has to be processed and exported out of the nucleus. So there are many steps that occur co-transcriptional. Okay? They are together with transcription. So imagine this is the RNA polymerase. And the tail, as I said, is very important because the tail will also recruit factors that are factors like protein that are also important for the processing of RNA. For example, here what we have capping factors. What these capping factors do, it's simply they are capping the 5' end of the elongating messenger RNA. Then you have splicing factors, which we are going to see in more details later. And you have polyadenylation factors. So all these are responsible for RNA processing. So first, capping. You can see that this is the messenger RNA that has been transcribed from the gene. And there is the coding sequence. This is the coding sequence that really is translated into protein. Okay, so there is amino acid code here. But in messenger RNA, there is as well a 3' non-coding sequence. It's only regulatory. It does not code for protein. There is the 5' non-coding sequence. And then there is the 5' cap. And you can see that the link, this is guanosine here, that is in addition methylated, all right? And it is linked to the messenger RNA by a triphosphor bond here. This is not encoded by the gene. This is added afterwards. In addition, on the 3' end, there is a polya tail that is added after transcription. So again, you will not see the polya tail in the DNA. This is added afterwards in the processing. And in more detail, this is the cap. So this is the first transcribed nucleotide, second and third. And then you can see there is a 5' to 5' triphosphate bridge, which is very different from this phosphodiester bond here. And then you have the 7-methylguanosine, because this is the 7th position here, and there is a methyl in this guanosine. So this is the 5' cap. The 5' cap is very important to protect RNA. In the cell, there are many proteins who are looking for messenger for RNA, and if they find a 5' end that is not protected by a cap, it will degrade the RNA. This is very important, for example, against viral RNA, which are sometimes not capped. So anyway, this is very important. In eukaryotes, protein coding genes are interrupted by non-coding sequence called introns. And this is where we are going into the splicing. In bacteria, you have a gene that is exclusively coding region. But in eukaryotic cells, here you have the promoter, and you can see that there are different parts. There are coding regions in red, which are called exons, and there are non-coding regions that are called introns. If you were to make this into a messenger RNA and directly translate it into protein, it would mean absolutely nothing. The protein will just not be functional. Before you translate this messenger RNA into protein, you have to remove the introns and you have to attach the exon together, which is a process called splicing. And some genes can have many exons, some genes can have just a few. For example, the human beta-globin gene, again, beta-globin, we have spoken many times about it. This is one of the subunits of hemoglobin. It has only 3 exons. And the human factor, this is one of the coagulation factors. Then you can see it has many exons, 26. So it can really be different. That being said, the length of a gene is actually corresponding to the number of exons. Some genes are very long, like in this case 200,000 nucleotide pairs, and in this case it's only 2,000 nucleotide pairs. Introns are removed from pre-mRNA by RNA splicing. So how can this happen? How can the cell know this part in the messenger RNA is intron and this part in the messenger RNA is exon? How can the cell know which part to remove? And this is again because of specific sequence within intron. So this is, let's say, the exon 1, and this is the next exon, the exon 2. Usually, the boundary between the exon and intron has a very specific sequence. which is usually this one here. And the intron-exon boundary, again, has a specific sequence. And in addition, there is something called a branching point, which is usually with this sequence. I don't know if you have seen these R, Y, N before. These are ambiguous nucleotides. They can be anything. So for example, in this case, Y is C or U. R is A or G. N can be any nucleotide. So this is what it means. So the cell will recognize this sequence, will know, okay, there is an intron here, and this is the end of the intron, so I should attach this exon there with the next one to actually reconstitute the major messenger RNA. And very simply, this is how it happens. So the ULAC, you remember the branching point? The central A is very important because the central A, there is a OH here that will be used for a nucleophilic attack on the end of the exon 1. And then you have an intermediate, which is called a lariat, just like a lasso or something. And then after that, because the bond here has been broken, then the end of the first exon will be used to attack the beginning of the next exon. So then you will create a new link between the two nucleotides there, and finally you have the major messenger RNA, and you have the lariat who goes away. And something that is very interesting is that the factors, shall we call them, the factors that actually mediate this splicing, you can imagine RNA cannot do that by itself, okay? It needs other factors. But the factors that are responsible for the splicing are RNA themselves. So you have the messenger RNA here. This is the pre-messenger RNA because there is still the intron here. And then you have splicing factors, which are mainly composed of RNA. They are actually called SNPs. Where is it? Here, SNPs. Small nucleolar ribonucleotide protein. Okay, so it's a mix between small RNA and proteins. And together, because they contain RNA, they will actually recognize the sequence in the intron, and they will actually mediate the splicing between the two exons. And something that is very interesting, I think, is that the catalytic activity is in the RNA molecules, not in the protein. There are proteins in the complex there, of course, but the catalytic activity that breaks the bond here and that attaches the bond between the exon is encoded by RNA. Okay, so it's a ribozyme. It's an RNA with an enzymatic activity. And so at the end, this lariat will be removed, complexed with the spliceosome. So the mix of all these, nuclear, all these, RNA that are responsible for splicing, complexed with protein, these are called the spliceosome. And this will go away. The lariat will be degraded, and then the messenger RNA will be spliced. But it's not always the same case, because the cell can choose which exon to include or to exclude. For example, in this case, you have a gene that is composed of 3 exons, and the pre-messenger RNA, of course, will contain all the exons, but after the splicing, you can either have 1, 2, 3, 2, 3, or 1, 2. And this is called alternative splicing, which means that one gene can actually code for more than one protein because of this splicing. This alternative splicing is not every gene like this. Some genes only have one pre-mRNA that is spliced into one major messenger RNA. But some genes can have a huge number of alternative splicing, okay, alternatively spliced messenger RNA. It really depends. And it has been observed that actually in the development of eukaryotes, the beginning of the mammals branch is actually alternative splicing became much more complex. So it is believed that the complexity of organism goes in parallel with how much alternative splicing there is in the cell. And it's easily understandable why. Because if you have one gene that can code for, say, 100 different proteins, then the complexity of the organism is much higher. Next point is mature eukaryotic messenger RNA are exported from the nucleus. Yes. So once the messenger RNA has been capped, spliced, polyadenylated, then it's associating with cap binding protein that's associated with the exon junction. It's called the exon junction complex, proteins that bind to the poly-A tail. And these proteins, complexed with the messenger RNA, will actually interact with proteins that are at the boundary of these nuclear pores. And together, they will mediate the export of the messenger RNA into the cytoplasm. But once the messenger RNA reaches the cytoplasm, then the protein will be exchanged, because the protein that interacts with RNA in the nucleus and the protein that interacts with RNA in the cytoplasm will be different. So it's extremely complex. There is a lot of research going on about this thing. But essentially, once the RNA reaches the cytoplasm, then it will be translated. And it's again many different proteins and ribosomal RNA that will interact with the RNA. Okay. Any question?
Just one question. Like the cap binding protein, after it has been replaced, like outside, it goes inside, right?
It goes.
Or what happens to it? Like
What do you mean? From the transcription? From this one?
yeah, after the protein exchange,
Yeah.
what happens to the cap binding protein? Does it like return to the nucleus?
Yes. So these cap binding protein, once they reach the nuclear pore complex, they will actually be, not all of them will be replaced. Some will be replaced, some will still bind. But essentially, if the protein is actually a protein that needs to be in the nucleus, then they usually contain small sequence that other protein will recognize to transport it back into the nucleus.
All right. Yeah, I was wondering, like, is it some kind of free exchange, but it's regulated?
Yes. So if for example this protein is in the nucleus, so in this case, once it's replaced by a cap binding protein that is a cytosolic protein, then this protein will then be transported back to the nucleus. It will not go by itself. It needs to be transported actively by other proteins that will recognize the sequence of that protein.
And I guess it can't escape alone from the nucleus as well.
It cannot escape alone. It needs to be associated with many different proteins. And something that you do not see here, which actually happens, is that this is not only one protein. Don't think that this is one protein. There are many proteins that interact with RNA. And actually, the proteins that interact with the poly-A, they interact with the cap binding protein. So it actually forms a very tight complex. Okay. And of course, the proteins that bind messenger RNA, some of them will be the same for all messenger RNA, but some of them will be specific for given messenger RNA, depending on the sequence of the exon junction complex, depending on the sequence of these non-coding sequence. So protein will not be constant. Okay? They will be different for each messenger RNA. Some of them will be the same, some of them will be different. Next point is mRNA molecules are eventually degraded in the cytosol. So we can see, I went a little bit fast, but the earliest cell may have had introns in the gene, so it's a little bit different. Let's first look at this one here. So that's the gene, that's the pre-mRNA, that's the major messenger RNA. then you have export, and then translation into protein. Degradation of messenger RNA, the book make it very simple. mRNA molecules are eventually degraded in the cytosol. Yes, of course. Some RNA are degraded very quickly. Some RNA are degraded not so quickly. Okay? It really, again, depends on what mRNA you are talking about. And degradation of RNA is, I think, extremely interesting because it can actually proceed differently. Some mRNA are degraded from the 5' end, so the cap is removed, and then RNA is degraded. But it can also be degraded from the 3' end, where the poly A tail gets progressively shorter, and then RNA is degraded from the 3' end. And there is a lot of research going on there because it seems to be really specific on the type of messenger RNA you are talking about. So this is very interesting. But the point is true, is that when RNA are in the cytoplasm, yes, they are translated, but they are also degraded. Okay? And this depends on every single messenger RNA. So you can see many different processes in eukaryotes. In prokaryotes, it's very simple. The mRNA is produced, and once it's produced, already translation can start. So translation will not wait. As soon as the messenger RNA goes out of the polymerase, ribosome can already interact with the messenger RNA and make protein. So all this is simultaneous. In eukaryotes, it is not. Okay? First RNA has to be made, then exported, then translated.
Is one mRNA translated only once by ribosome?
No.
No.
This is what we are going to see. And again, this depends on if you have in your mind one single molecule of RNA, some RNA are very good at recruiting ribosomes. Ribosomes will bind to RNA very strongly and will synthesize a lot of protein. But some mRNA are not so good at recruiting. So for one given RNA, one of them can make a lot of protein, one of them can make not so much protein. Okay? Now we are going to see, this is the idea of polysome. An mRNA sequence is decoded in sets of 3 nucleotides. Yes, so this is about protein now. So if you look at the sequence of the messenger RNA, of course it's composed, the coding sequence, we are talking about the sequence that is coding for protein. They are codons, which are a group of 3 nucleotides. And depending on the sequence of this nucleotide, this tells the cell to actually call or to add a specific amino acid. For example, this is alanine, GCA, GCC, GCG, GCU. And of course, there are 20 amino acids, so there is a little bit of redundancy. You can see 4 codons actually code for alanine. But what can you see? If you look at this very carefully, what can you see? What can you observe from this one? So amino acid, yes, of course, there is a 3-letter code, which is, I think, easier, and there is a single letter code. Some of them are easy. For example, alanine is A, arginine is R, aspartate is D. So it can be a little bit different, the single letter code. I usually prefer the 3 because there is no room for mistake. But anyway, take a look at the codons and take a look at the amino acids and tell me what you observe.
Redundancy is different based on amino acid.
Yeah, so for example, methionine has no redundancy. AUG is methionine. That's it. Arginine has a lot. This is one thing, that's true. And the reason why methionine is, it's a single codon, there is a reason that we will see later. What can you see in these codons? something funny is that also stop. Stop is not an amino acid. It just tells the ribosome stop translating. So this is a stop codon. We will see later. So what can you see? What can you see for this codon? There are 3 nucleotides, yes, but is there any pattern you can see? Especially, is the redundancy random?
Codons which translate the same amino acid has the same first 2 letters.
Yeah, exactly. So not every position is as important for the coding into amino acids. So for example, let's check this leucine here. You can see that leucine can be UUA or UUG. The second nucleotide is always the same, but it can also be CUA, CUC, CUG, CUU. So the last position, the third nucleotide of each codon, is actually not very informative. Okay? This is called the wobble position. The first 2 are very important, but the last is less important. Okay? Again, here you can see glycine here, GGA, GGC, GGG, GG. So the first 2 are important, the last one is less important. All right. How do we know this? How do we know these things? Nowadays we take it for granted when we do experiments. But actually, maybe 40 years ago, people didn't know what was the genetic code. So how did they know? I think it's pretty interesting is that they actually started by making RNA molecules that contain only 1 nucleotide. So that was very easy for UUUUU. It was very easy for AAAAA. It was very easy for CCCCC. But it was not easy for GG, because if you make an RNA molecule that only contains GG, it makes a lot of very hard secondary structure, and it's impossible to use in this experiment. But now, they could determine the first 3 codons, and what they were doing, they were synthesizing this fake messenger RNA, and then they were putting it into a tube that contained cellular extract, ribosomes and everything, and they used some maybe radioactive substance, and then they could see that a radioactive protein that was made from this synthetic messenger RNA only contained the phenylalanine amino acid. Yes, if you take a look, UUUUU, this is phenylalanine. So this is very easy. And if they do the same thing with AAAA, this is lysine. So that's very easy. The problem is when they had to know about other codons that are made of more than 1 nucleotide. For example, poly-UG. So you have UG, 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 UG. In this case, this is not so good because UGU and GUG, it can be either cysteine or valine, but they don't know which is which. So that's a problem. Then they had poly-AG, so they have AG, 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 so it can be arginine, glutamate, arginine, glutamate, and so on and so forth. Poly-UUC, that's even more difficult, because it can be either phenylalanine, 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 or serine, 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 or leucine, leucine, leucine. So this is very difficult. They did not know how to do it. And the technology to actually make a complex RNA molecule containing more than 1 type of nucleotide did not exist at that time. Still, they had to do it. And actually, they managed because a different scientist at more or less the same time, I forgot his name, it's in the book, but a different scientist actually managed not to make RNA, but managed to make DNA. Because the chemistry of DNA is a little bit easier than the chemistry of RNA, then scientists knew how to synthesize a DNA molecule of a specific sequence. So what the idea they had was first making a DNA molecule of a specific sequence, then transcribe it in vitro using RNA polymerase. So they could make a copy, an RNA copy of the first DNA sequence, and then they could do these things. Okay, so this is how they made the first step. But again, even though they could make an RNA molecule from a previously synthesized DNA molecule, they could not know exactly which codon corresponded to which amino acid. And it's only later that they actually managed, and they noticed that if you only make an RNA molecule containing only 3 nucleotides, only 3, this is enough for a ribosome to bind to it and call a tRNA and match a transfer RNA with that specific sequence. And they could actually see that when they only use UGU RNA molecule, they can actually see that what they have with a UGU is this one. I don't know them by heart. I don't ask you to know them by heart. UGU is cysteine. Only using small string of 3 nucleotides. So they could precisely know, okay, so if we synthesize this, then we can see that the tRNA we purify from this actually bears the cysteine amino acid. So this is how they could tease apart the code. First, by doing a strand of single nucleotides, then by making a mix of different nucleotides, and then by using only a trinucleotide mRNA sequence. Anyway, there is one more problem. And this is very important in our set, because if you consider one RNA molecule, which is CUC, whatever the sequence, depending on which amino acid, which nucleotide you start with, you will synthesize a completely different protein. This is called the reading frame. In this case, the reading frame starts from the first nucleotide, and this means leucine, serine, valine, threonine. But there is another reading frame which actually starts from the second nucleotide, and in this case it's serine, alanine, leucine, protein. Completely different. And there is a third reading frame which actually starts from the third. So this is very important, and you can imagine for ribosomes inside our cell, it's very important to know where to start. Because you can make a protein that is very functional, or you can make a protein that is completely rubbish. Okay. So this is very important. So let's see a little bit on how the cell is decoding nucleotide into amino acid. tRNA molecules match amino acids to codon in messenger RNA. This is simplified structure of a transfer RNA or tRNA. And it has more than, it's usually likened to a clover leaf like this, or a shamrock if you want. And there are different parts, but I would say the two main parts is the anticodon here, which is actually the part that will bind by hydrogen bonds to the messenger RNA molecule. And at the other end of the tRNA, there is the amino acid that is attached here at the 3' end of the tRNA. In space, this is the structure of tRNA here. Here in blue, this is the anticodon, and here this is the attached amino acid. So you have to be careful, okay? It's called anticodon. So if I ask you, what is the codon that is recognized by this tRNA? You tell me what? What is the codon sequence that is in the messenger RNA that is recognized by this phenylalanine tRNA? This probably is a similar question you get at the exam as well. I love this question. So this is the anticodon.
UUC.
UUC. UUC. Because here, 5' end, and you get GAA. So it means that the messenger RNA it will pair, but in the other direction, antisense, right? This is 5 to 3, so the messenger RNA will be 5 to 3, so that will be UUC. Okay, well, we'll see this one later. So this is tRNA, and tRNA are very important because tRNA will assemble with ribosomes together with RNA, messenger RNA, and together will synthesize protein. Okay. So this is just a bigger picture. Specific enzymes couple tRNA to the correct amino acid. It is wrong to say that only tRNA are important for matching a codon with an amino acid. Actually, these proteins are extremely important as well. For each tRNA, there is a specific protein that will match a specific tRNA with a specific anticodon with its corresponding amino acid. And this is a tRNA synthetase. So it's an enzyme that synthesizes tRNA, complete with the tRNA, the tRNA molecule, and the amino acid. So two things are important. Not only tRNA, but these proteins that actually synthesize each of the tRNA. If there is a mistake in this tRNA synthesis, there will be a mistake in translation. And what this protein does, as you can see here, is actually make a covalent bond between the amino acid here and the tRNA molecule. And here again, so this is a bit easier than here, but you can see that the anticodon of this tRNA is ACC, and the codon in the messenger RNA is actually GGU, but it's very difficult. Again, they are a little bit difficult here, because they actually show 3' to 5'. The convention in nucleic acid is always to write, to read and write from 5' to 3'. Okay? So normally, the anticodon here will be CCA. If you read from 5 to 3, okay? CCA. And the codon, UGG, this is correct. This is in the correct direction. But ACC, it's not the correct direction, okay? ACC. Because you can see in this case, ACC, and you get tryptophan, okay? Let's take a look. Where is ACC? Where is ACC? Here. Asparagine. See, this is different. Because all these codons are shown from 5 to 3. 5' to 3', which is the convention. On this figure, be very careful, because the sequence is shown from 3 to 5. So you have to be careful. Okay, look.
You said AAC.
Yeah, I mean, this is ACC, but this is not correct. The codon for the messenger RNA, UGG, this is correct, but CCA is not correct. ACC is not correct. Okay, look.
Which is not correct and
This
correct.
UGG is correct. UGG is the codon coding for tryptophan. But if I ask you, what is the anticodon of this tRNA, what is the correct answer? If I ask you, what is one of the codon coding for tryptophan, then you tell me UGG. I tell you, fine. But if I ask you, what is the anticodon for the tryptophan tRNA, what do you tell me?
CCA.
Yes, CCA. Because the convention is from 5 to 3. But on this picture, they show 3 to 5. So they are trying to confuse you. Okay? There will be questions like this in the exams. I know.
It is nonsense to find ACC in not anticodon in codon table.
Yeah, so you don't usually have anticodon table. What you get is codon table, like you have this one here. This is a codon table. So this is the 5' to 3' sequence in the messenger RNA. It's not the sequence in the tRNA. Okay, so you have to do a little bit of mental gymnastics there. All right. The messenger RNA is decoded by ribosomes. So you can see here, this is the endoplasmic reticulum, as we have seen before. And some of the ribosomes are apparently free-floating in the cytosol. Some of them, many of them, are actually tightly associated with the endoplasmic reticulum. And this is the site of translation. And probably the biggest complex in the cell, one of the biggest complexes in the cell, is this ribosome. Ribosomes are made of two subunits. The large subunit with a very huge molecular weight of about 3 million or something. Then you have the small subunit, which is about half the size. And it's mainly composed of ribosomal RNA and protein. Okay? About 49 ribosomal protein plus 3 RNA molecules. These RNA molecules are huge. Okay? They are very big. And then the small subunit is again a ribonucleoprotein molecule. Okay, so it's a huge complex, and the two subunits actually associate into one ribosomal unit, which is shown here as a molecular model. And there are 3 sites in ribosomes. There is the A site, there is the P site, and there is the E site. The A site stands for acceptor. It's easy to kind of understand why they are named like this after the next slide. So this is the acceptor site. This is the peptidyl site, or peptide site. And this is the exit site. Okay? 3 sites. And these sites will actually bind tRNA. At the interface between the small and the large subunit, this is where the mRNA will fit. Okay, so let's see how it works. So you imagine a messenger RNA molecule here. Do I have it bigger? No, I don't have it bigger. So this is the messenger RNA molecule here, and that's the step 1. So there is already a tRNA who is in the P site. And then there is a new tRNA that comes into the acceptor site. The second step is the 2 amino acids here will be attached, and the link between that tRNA and that amino acid will be broken. So this is what happens here. Then what happens is that the large subunit will translocate to the 3' end, like this. So this will actually shift this tRNA to the exit position, and this tRNA to the peptidyl position, as you have here. Then the small subunit will also translocate to the 3' end, which will cause the tRNA in the exit site to go out, like this. And then the step 1 again, where you have a new tRNA that comes into the acceptor site. And this is just repeating the same thing over and over again. And this is a film that shows that exactly. Maybe I can show just to make sure. This is the protein that is loading the tRNA onto the ribosome. If there is a mismatch, it's not good. It's taken away and replaced by a good tRNA. This is a good match. So then the loading protein will be kicked out. And then, yes, come on. There you go. Large subunit shift. These things need energy, so this is why you have all of the proteins. So there are different proteins that load the tRNA, and there are different proteins that actually make sure that the subunit of the ribosome shifts. Okay? They don't shift by themselves. Protein make them shift. Okay. And that's the same thing, not with the RNA polymerase, but with the ribosome or ribosome. So this is the RNA going out of the nucleus. You can see the nucleus with the pores there. This is not correct. RNA doesn't look like a ramen noodle. Okay, it's tightly packed with protein. But then essentially you have the small subunit, the large subunit, and all the tRNA cones, and it looks like a typing machine. And then tRNA are adding one after another, and after some time you have the peptide that actually leaves the ribosome. And we can see each of the tRNA with their little amino acid attached in red. Now, this is moderately interesting. Okay. So ribosomes are huge, but there are many proteins, but in terms of mass, RNA is the main thing. Okay? And the ribosome is called a ribozyme. So I have said this word before. A ribozyme is an RNA that has a catalytic activity, just like an enzyme, but it's not a protein, it's an RNA. Okay, please remember this one. This is ribosome. This is the RNA that composes a ribosome, and you have small ribosomal subunit, which is 5S RNA, and you have the 23S RNA. So it's a huge, huge RNA molecule that has catalytic activity. Specific codons in messenger RNA signal the ribosomes where to start and to stop protein synthesis. So this part is very interesting because you imagine this is the cap of the messenger RNA. How will the ribosome know where to start translation? Because I told you, the reading frame is very important, because if you make a mistake, you will make a wrong protein. So what actually happens is that you always have something, okay? It's a translation initiation factor, which is a protein, together with a small ribosomal subunit. They will bind the cap at the beginning of the messenger RNA. And this small subunit is associated with a specific initiator tRNA that has a methionine in it. It will bind the cap and it will go down in the 3' direction until it meets the codon AUG, which is a match for the anticodon of this initiator tRNA. So once the initiator tRNA binds to its codon, then this will be the site, okay, I have reached the translation initiation site, I should start translating. And then the large ribosomal subunit will be recruited and translation will start. Okay. And this is all due to a very specific sequence, which is the AUG translation start site. Where to stop, that's the same thing in bigger, okay? Which means for this is a messenger RNA, you will tell me in which animal it is or which living thing it is. So this is, imagine a messenger RNA, and in this case you have 3 ribosome binding sites where immediately downstream there is a AUG, 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 which signals the start of a protein. So here we have a messenger RNA that codes for 3 proteins. What kind of living thing this is in? What do you think? This kind of messenger RNA. What are we speaking about here? Are we speaking about bacteria or are we speaking about eukaryotes?
Eukaryote.
Hmm?
Eukaryote.
No. This is not in eukaryotes. This is in prokaryotes, because this is what is called a polycistronic messenger RNA. In eukaryotes, there is only one protein for each messenger RNA. This is different from splicing. Okay? This is one messenger RNA that codes for 3 different proteins translated from 3 different initiation sites. In eukaryotes, it's a little bit different. Okay? In eukaryotes, you usually have one single translational start site and one single translational stop colon. Okay, so this is in bacteria. How is the stop signal given to the ribosome? Well, this is exactly similar to what happens in the... Do I have it in here? In this case, the stop codon, as I told you before, is UAG. It can be also a different, it could also be, as you can see here, there are 2 different stop codon. You have them in the codon here. It can be UAA, UAG, or UGA. They do not have the same power. Okay? Usually when I make a bit of DNA that codes for a protein that I want my cell to synthesize, I always use TAA, UAA, because it's the strongest. It's a very, very, very strong stop site. Anyway, so they are not exact, they are not really identical. They have different power. But what happens here is essentially once you have the stop codon here, it actually recruits a release factor that will bind in the acceptor site of the ribosome. It will change the structure of the ribosome so that a link will be made between the last amino acid and water instead of between the last amino acid and a new amino acid. And then this is the end of the peptide chain. Once this happens, then translocation, and since there is no new amino acid coming, it's dissociated. This is how translation stop is signaled to the ribosome. Proteins are made on polyribosomes. So this is your question earlier on. This is usually what you have. And this is a model of what you can see in the electron microscope, where you have one single RNA molecule from the 5' end to the 3' end that is being read by many different ribosomes. So simultaneously, we have here 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13 ribosomes who are reading at the same time the messenger RNA. So simultaneously making so much more copies of proteins from that messenger RNA. So this is called a polyribosome. Next point is inhibitors of prokaryotic protein synthesis are used as antibiotics. Yes, you can imagine that if you want to kill bacteria or if you want to kill something that is alive, what do you think? You can inhibit DNA synthesis. You can prevent replication. You can prevent transcription. Or you can prevent translation. So usually you have 3 choices. What is the advantage of using inhibitors of protein synthesis? In this case, of course, prokaryotics, antibiotics. If you have a bacteria, if you have an infection, then you are using an inhibitor that will specifically inhibit the translation in bacteria. What is the advantage of using translation inhibitor, let's say, versus an inhibitor of DNA synthesis?
You can kill cells which is not dividing.
Yes. So for example, if your cells are not dividing, using DNA synthesis inhibitor, nothing will happen. But that's not all. That's not all. Because usually bacteria are multiplying. Okay? If you inhibit DNA, you will you can heal as well. But what is the advantage? What do you think might be the advantage of inhibiting protein synthesis over inhibiting DNA synthesis? Well, it's linked to what you say. It's much quicker. It's much quicker. If you inhibit protein synthesis, then every single bacteria in what you are giving antibiotics to will be killed, regardless whether they divide or not. It's much quicker acting. Eventually, if you use a DNA synthesis inhibitor, you are preventing the bacteria to multiply, so at the end you will heal, because your body will start killing the bacteria or whatever. But if you use protein synthesis inhibitor, then every single bacteria in your body, for example, will die because they are unable to synthesize their protein. And because the machinery that synthesizes protein in bacteria is different from the machinery that synthesizes your protein in your body, they are different, then this is why it's usually convenient to do that. So these are some very often used antibiotics that we can also use here in the laboratory when we do experiments, where we have tetracycline, streptomycin, chloramphenicol, cycloheximide, and rifamycin. And you can see they have different actions. For example, streptomycin is very well known. It actually prevents the transition from the initiation complex to chain elongation, also cause miscoding. So this will cause a lot of mistakes, so essentially the bacteria will die. Tetracycline blocks binding of amino acid tRNA to the A site of the ribosome. So this is something that definitely inhibits translation, and so on and so forth. Okay. So very often antibiotics inhibit translation. The next point is control protein breakdown helps regulate the amount of each protein in a cell. Yes. Usually, the first idea that everybody has is that you regulate the amount of protein you have by regulating translation. But regulating how quickly you break down your protein is also very important. And of course, there is a specific machinery in the cell that breaks down protein. And this is called the proteasome. And the proteasome is again a huge multi-subunit protein complex whose sole purpose is to degrade protein. But don't degrade any single protein without checking what it's degrading. You actually need to have a protein that has been modified by a small peptide called ubiquitin. Okay. So here we have 4 ubiquitin that have been added to this target protein. And once the target protein is ubiquitinated, then it is recognized by the proteasome and it is broken down into single amino acid that can then be recycled to make other protein. And ubiquitin is just like this. It's a very small polypeptide that is added onto protein to be degraded. So it's just like a label. Just like in Japan, when you have a big rubbish that you want to trash, if you put it outside your house, it will never be taken. If you want to get rid of it, you need to go to the company and you need to ask for a label. I have this big rubbish that I want to trash. Then you need to put a label and then you have the people from the city who will come, they will see a label, and they will take it away. So this is exactly what happens here. Ubiquitin is a label for the cell to recognize that this specific protein is to be degraded. And it's polyubiquitin because not only one ubiquitin is necessary, but more subunits of ubiquitin is necessary. For example, the age of the protein is correlated with how many ubiquitin it has. The older it gets, the more ubiquitin it accumulates, and the cell will recognize, oh, this protein is very old, we have to remove it, we have to degrade it. So that's the degradation of protein business. So that's almost the end of this chapter. There are many steps between DNA and protein. It's a kind of a synthesis of what we have seen today. So you have the gene here that of course contain exon, intron, and start site, and so on and so forth. Then you have the, transcription, 5' capping, elongation, splicing, then maturation of the RNA, export. RNA can be degraded, so this is a control of gene expression as well. How quickly you degrade RNA has an effect on how many proteins you make at the end. Then you have translation, and then you have protein degradation, and so on and so forth. In addition, the protein that you make by itself may not be functional. The protein might need also to be modified, phosphorylation, methylation, many other chemical modifications, and associate with different peptides to make the final functional protein you are talking about. So there are many potential points of control of gene expression or protein function. The cell can act on each of the steps to regulate how many final functional protein you have. Then the last point is something that I kind of like this one. It's about the origin of life. So I told you before that it's currently thought that RNA is the beginning. Why? Because if you consider the central dogma, DNA to RNA to protein is great, but you need DNA that codes for protein. So you need to code for protein, but you need protein to synthesize DNA, and you need protein to make RNA, and you need protein to translate. Which is first? It's just like the chicken and the egg. Okay? It's like the chicken and the egg. So which came first in the history of life? And now, some people think proteins. So yes, proteins are great because they have enzymatic activities. But so far we know no example of protein that is able to code for itself. Amino acids have no coding power. Okay? DNA has coding power. It can code for itself, but it has no enzymatic power. It's very stable. This is why we have DNA in our cells, because as a keeper of genetic material is great. So the only thing that could actually do both is RNA. RNA is able to code. It has a genetic code. But as we saw for the ribosomes and for all these RNA that are responsible for the splicing, RNA have enzymatic activity. So RNA is the best choice for the origin of life. Okay? That means that at the very beginning, RNA, well, it's only a speculation, but RNA might have been the very first molecule able to make more copies of itself. Okay? Because that's what life is. It's autocatalytic activity. Okay? So that's brief history of life here. So life occurred shortly after 5 billion years from now. And this is believed to be the RNA world. So it's not really the beginning of life, but the first cells with DNA are maybe thought to arise about this time here. But before that, that was a world only composed of RNA. Why? Because RNA molecules, ribose, the sugar that actually is inside nucleotides, it can be made spontaneously from inorganic molecules. So if you have formaldehyde, do you know formaldehyde? formaldehyde, it's a very simple, very reactive molecule that is, but there are proof of that, but that's believed to be very abundant in the primal Earth. So at the very beginning, when there was nothing alive yet, there was a lot of formaldehyde on Earth, and actually formaldehyde can spontaneously form ribose. So there are many kind of like observations like that that suggest that there are a lot of reasons why RNA could actually be the first molecule on Earth that had life-like ability. And life requires autocatalysis, I told you before. RNA can both store information and catalyze chemical reactions. So this is the coding of information I told you about. So if you imagine the original RNA, due to this base pairing, it could actually make its complementary and could use after that the complementary to make a copy of itself again. Okay, so this is kind of how one single strand of RNA can actually catalyze its own synthesis. But then there are scientists who are very interested in that, and they have made in vitro RNA that have been, if you want, selected in vitro. So they try to reproduce what time could have done with RNA molecules, and they did that in the laboratory. So for example, peptide bond formation in protein synthesis, you know, this is something that already exists, that the ribosomal RNA. But scientists have made pure RNA molecule that can actually do DNA ligation. So this is in vitro selected RNA. RNA splicing, you know, this actually exists in the cell. But scientists have made RNA molecule that can polymerize different molecule of RNA, or some RNA that can phosphorylate RNA, RNA that can add amino acid to RNA. So all these in vitro selected RNA, they are RNA molecules with catalytic activities that have been synthesized in the lab. Okay? And then you have more other, like for example, RNA alkylation. You know what is alkylation? Alkylation. What is alkylation? You know what is phosphorylation, polymerism. You know this one, but alkylation, what is alkylation?
Attaching alkane.
Alkane, so it can be like, the smallest is methyl, CH3, but it can be ethyl, CH2, CH3, not just C and H. Okay. So all these RNA have been made in vitro, and this is a kind of indirect demonstration that yes, the possibility of RNA molecules is very large. You can make RNA molecules that have catalytic activity. So for example, this one here, you have an RNA molecule that will actually break, that will cause a nick in a different substrate RNA. So you can imagine that many RNA molecules with some sort of catalytic activity able to maybe autocatalyze their own synthesis, and they will be submitted to the competition with other RNA molecules. They will compete with other RNA molecules for molecules that are present in their environment. So this is really kind of beginning of how life could have arisen. Well, this is just the same idea here. And the last point of the chapter is RNA is thought to predate DNA evolution. So that's currently one of the most accepted theory, is that in the beginning it was only RNA-based system. Then in the course of evolution, then RNA started to sort of code for protein, and protein, because they are much more variable, 20 amino acids versus only 4 nucleotides, there is much more possibilities in proteins. So they represented an advantage for these pre-living organisms. And then after that, then living organisms kind of invented DNA as a better molecule to keep genetic information. And then there was specialization of DNA as a keeper of genetic information, protein as enzymatic activity, and RNA has not completely disappeared, because still in our cells there are many RNAs that are necessary for life, that have catalytic activity, and that are encoded in DNA. Now, this is just believed to be the original. And then we have seen this one before, that DNA compared to RNA is better because you can actually easier recognize when you have a mistake. For example, when you have deamination of cytosine to uracil. In DNA, you can notice that immediately. If you have that in RNA, then it's very difficult. So RNA molecules are much more fragile. So DNA represents a kind of evolution. Anyway, that's the end of the chapter. So if you have any question, please fire away. Otherwise, I will see you next week. I went fast today. I went very fast. So if you have any questions?
Next week, will we have, what will we have?
Next week, so this is chapter number 7, right? So next week, I actually want to go over chapter 8, 9, and 10, and then finish at chapter 10 for the exam. So I want to see 3 chapters next week.
Oh, very fast.
This will be very fast. It will be very fast. No, it's not true. It's not true. It's a stupid joke. What I probably advise you is to read the book for next week. And then if you have any questions, then fire away. Okay. We can discuss about stuff. If there are things you don't understand in the book, please ask. But I will not see anything new. It's just too late. Okay.
Chapter 8 to 10?
No, that was a joke. That was a stupid joke. So I will not see any new chapter. But we can just, read the book. If you have any question, please ask. Okay. What we can do, for example, is go over the answer that you There are questions that you have, right? At the end of each chapter. Try to do them. And if there is one you don't understand, then you can come and explain and ask, for example. That would be a nice way. Okay, for example, question 7.2. In the electron micrograph in figure 7.8, are the RNA polymerase molecules moving from right to left or from left to right? So you can do these questions by yourself. Okay? That's one video. So anyway, but I will not see a new chapter. This will not be fair. Okay, that's it.
See you next week.
See you next week.